In this video, I'm going to talk a bit about what's often called gain-of-function research, which you will often see abbreviated as GOF research, because this has come up in the news, particularly because of the heated exchange between Dr. Anthony Fauci and Senator Rand Paul. And it's essentially looking at whether the United States, the NIH, the National Institutes of Health of the United States, funded so-called gain-of-function research, particularly at the Wuhan Institute of Virology over in China, where the coronavirus, where SARS-CoV-2 is thought to have first come from. And so this video is largely sort of informed by what I've looked into for that, and from a conversation I had with a commenter on my video about the lab leak hypothesis. I believe their screen name was Nathan's HVAC. But anyway, he linked me to a video that was quite informative. But anyway, so gain-of-function research, or is sort of a slippery term, and it when I sort of started really thinking about it, it kind of threw me for a loop for a day or so, because you could kind of construe a lot of research as gain of function, because a lot of the what's called recombinant DNA techniques that are used in pretty much every laboratory where you do biochemistry or cell biology or microbiology or anything like that does recombinant DNA techniques. And you could almost construe, if you define gain of function research too broadly, you could sort of encompass a lot of sort of standard everyday lab techniques, whereas if you defined it too too broad, then you would end up having recombinant, so recombinant DNA techniques being sort of sucked into the gain of function, which has a negative connotation to it as something that is dangerous. But if you define it too narrow, then you might miss some potentially dangerous sort of experiments. And so we could maybe even do this as an illustration. So if we say that this is the space of all recombinant DNA techniques, we might want to say that gain of function, so we'll say that this is all recombinant, and this down here is gain of function. And so it's a matter of how wide or how narrow we want to define gain of function. And so the video that the commenter linked me to was informative because it gave sort of more concrete standards for how we might want to define gain of function, so that it doesn't start encroaching too much into all this other recombinant DNA techniques. And so the video essentially gives three things that we would want to, so I'll kind of block it off from over here, three things that we would want to consider when we are talking about gain of function, or what is oftentimes called potential pandemic pathogen research, or PPP research. And so in the video they talked about that you would want to look at the novelty of what you are creating. And this is important because this could pose issues for our immunity. So if it's something that's too novel, then we would not have any sort of pre-established immunity based on other pathogens that we have encountered. And so the next one is the pathogenicity. The pathogenicity, which is essentially how much this, well, pathogenicity is how dangerous the pathogen actually is. So essentially the death rate. So that is the pathogenicity and transmissibility. So transmissibility, which is how much it can spread. So you may have encountered, if you've looked into this, what's called the R0 or R0, which is essentially if one person, so if person 1 has the disease, the R0 is how many other people that person could spread it to. And so if you have if you have P2 and P3 here, if P1 spreads it to only P2 but not P3, you might say that the R0 is somewhere around 1. If you have R0 less than 1, then that's saying that this person has a less than 100% chance of passing it on to one other person. So if you have a bunch of people, say you had 100 people, then you would have maybe only, like if R0 was equal to 7, then only 70 out of 100 people would pass it on to another person. Whereas if you have an R0 greater than 1, then you could have P1 passing it on to more than one person. So for instance, if you had 100 people, they might pass it on to 120 people if R0 was, say, 1.2, for instance. And so that is sort of the R0 is that transmissibility of the pathogen. And so so we can define gain of function in the sort of negative connotation sense, or the potential pandemic pathogen research as research that would introduce one of these things, or increase, I guess you could say, one of these things in a pathogen. So it could increase the novelty, which would be something like giving pathogenicity or transmissibility to a virus that did not have it before. So if you gave pathogenicity or transmissibility to a virus that humans had not encountered before, that was not a virus that infected humans, that would be an example of novelty, where pathogenicity is something you could increase by taking a virus that humans can catch, that can infect humans, and making it more dangerous, so increasing the death rate that it causes. Or you can increase the transmissibility, which would be essentially increasing this R0 here, so increasing the number of people that it could, that a person infected with it could then infect. And so one way of doing that would be making essentially a virus that is not airborne into one that is airborne. And so that would be a way of increasing transmissibility. And so I guess you could, if you, an example of a virus that is high pathogenicity but low transmissibility would be something like Ebola. So Ebola, it's not, it is transmissible, obviously, but it is not highly transmissible, but it is highly pathogenic. It has a high death rate. Where I guess you could say that something like the flu, sort of the yearly flu, is something that you would say has a high transmissibility, but not too high of a pathogenicity. I mean, not compared to like Ebola or something like that. And so that is kind of the difference between those two things. And so that is something that I guess kind of helped me think about this from that video, which I will have linked to in the description down below. And so the video is talks by two different scientists. The first one, I guess, is somewhat more pessimistic about potential pandemic pathogen research, in that he runs through some calculations that show that the way we're doing things, or at least the way we were doing things in 2015, which is when the video was recorded, is potentially quite hazardous. So he runs through some numbers showing that it's quite hazardous. The second person in the video to give his talk is a bit more optimistic and thinks that we should be doing more potential pandemic pathogen research. I was going to run through the numbers that the first guy in the video did, but I think I'm not going to do that. You can watch the video if you're interested in that. Also, I've found his PowerPoint slides online, and I'll have that linked to below. That's where I copied and pasted this stuff from. But essentially, the pessimistic guy concludes that the risk, which is equal to the probability of something happening times the consequence if it happened. So he runs through the numbers, and he ends up with this here in blue. So between 2,000 and 1.4 million fatalities per biosafety level 3 labs in a this lab year here is essentially a lab running for a full year. So a biosafety level 3 lab running for a full year, using numbers that he gets from these these papers here and these papers here. But anyway, so 2,000 to 1.4 million fatalities per BSL-3 lab year, using the numbers that he got. But anyway, and he talks about too that there are ranges here for all his percentages. But anyway, I'm not going to go through that. I will talk a little bit about the actual paper in question in the Dr. Fauci, Senator Rand Paul controversy, which is this, and I'll have this linked to in the description down below. So this paper right here, and so this is copied and pasted from that paper, from the relevant portion of that paper. And I'm going to sort of go through a little bit here what the authors of this paper actually did. So I'm going to read through this. So in the current study, we successfully cultured an additional SARS-CoV, which I'm putting these in red here are my words, not from the paper. So a SARS-CoV is a SARS-related coronavirus. So it's a coronavirus found in bats, essentially, that have a lot of sequence identity to the SARS coronavirus. And this paper is from 2017, so it's from before the COVID-19 pandemic. So they are looking at the SARS-CoV-1 from the early 2000s. So they are looking at these viruses that they get from taking fecal samples from bats. And so these numbers, these alphanumerical designations here are the SARS-related coronaviruses that they find in the bats. I think the RS, so the letters in it tell you what kind of bat it is from, and the numbers are just sort of a designation for that virus. So in the current study, we successfully cultured an additional novel SARS-related coronavirus, RS-4874, from a single bat fecal sample using an optimized protocol and Viro E6 cells, which are a type of so they're a cancer, a human cancer cell. I think they're kidney cancer cells, but they're used essentially because they are, well, they're immortal, and so you can just keep using the same cells. Well, you can keep using cells in that lineage anyway. But anyway, that's what those Viro E6 cells are. So this RS-4874 had its spike protein, its S or spike protein shared 99.9% of its amino acid sequence identity with that of the previously isolated WIV, which is Wuhan Institute of Virology 16, and as well as identical to the WIV16 virus in the receptor binding domain. So they used a reverse genetics technique, which I don't know how much I want to get into it. I will leave a link to the paper that they sort of link to here down below. But essentially, what it is that the coronavirus genome is kind of really long, so it's hard to sort of I guess work with all in one large sequence. So what they do is they sort of break it up into multiple sequences, and then they insert just parts of a sequence into, so if we say that this is part 1, 2, 3, 4, so they put part 1 into what's called a bacterial artificial chromosome, which is just a type of plasmid, so a small ring of DNA that you can put into bacteria in order to express it, and then get the protein, or you can have it, or you can increase the amount of that plasmid that you want by letting the bacteria replicate. But what they're doing is essentially putting one part into a plasmid, then part 2 into another plasmid, and then part 3 into another plasmid, and part 4. And I don't know how many offhand, how many sections they actually, how much they actually sectioned this off into. But this is essentially kind of what they're doing, is just taking parts of the genome and putting it into different plasmids, and then they can actually they can take the bacteria that these plasmids are in, and they can actually sort of cut these sections out and then sort of recapitulate the entire genome once again to sort of recover the virus. And so that is what this reverse genetics technique is that they're talking about. So they constructed a group of infectious bacterial artificial chromosome, which is what these are right here, that I was just talking about. And these bacterial artificial chromosomes allow for what's called conjugation, which is just supposed to help sort of Well, it's supposed to make more efficient the sort of transmission of these plasmids between bacteria as you allow those bacteria to um, to replicate in order to increase the copy number of each of your plasmids. So they put this. So they put these SARS-related CoV clones into the backbone of the Wuhan Institute of Virology 1 or WIV1 backbone. And so what that's essentially saying is they have a plasmid, and so I'll have it broken up here. So you would cut that with restriction enzymes here so that it linearizes the circle. And this right here is the WIV1 backbone. And then they take the spike protein, which they cut out of the genome for the virus. So just the spike protein, and then they put that into here so that you end up with a plasmid that has the WIV1 backbone with the spike protein from one of these other SARS-related CoVs. All right. So anyway, I'll go back and reread part of this. So using the reverse genetics technique we previously developed for WIV1, we constructed a group of infectious bacterial artificial chromosome clones with the backbone of WIV1 and variants of S spike protein genes from 8 different SARS-related CoVs. So only the infectious clones. So these two variants here led to cytopathic effects, essentially causing damage to the cells, i.e., causing illness in their Viro E6 cells after transfection, which is just adding the virus onto sort of a petri dish full of your cells, so the Viro E6 cells. So the other 6 strains with deletions in the receptor binding region of the spike protein failed to be rescued. In other words, they failed to infect the cells. But in contrast, when the Viro E6 cells were respectively infected with the 2 successfully rescued Chimeric, so the WIV1 backbones with these SARS-related CoV spike proteins had efficient virus replication was detected in all infections. To assess whether the 3 novel SARS-CoVs can use human ACE2 as a cellular entry receptor, we conducted virus infectivity assays with the expression with or without the ACE2. All viruses replicated efficiently in the human ACE2 expressing cells. So the takeaway part here is this idea of taking the WIV1 backbone, and in fact I'm going to talk about it in the linearized sense because we have the genome here. So this is the WIV1 genome, and say that this part is the spike protein, so the spike for the WIV1. And what they're doing is they cut this part out so that they have the WIV1 without its spike protein here. So WIV1, and we'll just say BB for backbone. And then they take the spike protein from a different coronavirus, so we can say plus, the spike protein from a different coronavirus and add that into, so S for some other coronavirus, and add that into the genome here. So now we have this spike for the other coronavirus in our WIV1 backbone. And so I guess the idea that's in question here is whether taking the spike protein, which is the protein needed for a coronavirus to enter the cell and infect the cell. So taking the spike protein from one and inserting it into another one, is that gain of function? So does that satisfy the criteria of novelty, pathogenicity, and transmissibility. So transmissibility. I mean, it doesn't confer that much novelty because it's taking just one natural plus another natural. So the spike protein is not being altered in some way. It still has this naturally occurring spike protein. And by the way, the WIV1 is able to infect humans. And so what they were looking for is whether this other spike protein was able to infect humans. What their experiment wanted to show is if they took this WIV1, which can infect humans, and inserted this other spike protein, if it did not then infect humans, then they would say that this other spike protein was not infective to humans. They did indeed find out, though, that it can use human ACE2 as a cellular entry receptor, and so it was infective to humans, but the WIV1 was already infective to humans, was already able to infect humans. And so I guess then what you would want to know is whether adding this other spike protein, which was still able to infect humans, did that increase the pathogenicity or the transmissibility of this new chimeric coronavirus that had the WIV1 backbone with this other spike protein. So that's something that you would need a different kind of experiment, I think, to tell whether that was the case. You would need to do it into an animal model in order to assess whether it increased pathogenicity or whether it made it more transmissible, because this was done only in cells, not in animals. At least as far as I remember, I don't think the paper did any animal model, any in vivo studies with these new chimeric versions. So I think the issue here is that this could easily be construed either way, as a sort of quote-unquote gain-of-function experiment, or as not being a quote-unquote gain-of-function experiment. So just given the results in this paper, I don't know if we could say that. I mean, you could maybe determine from the cell culture experiment whether it was more pathogenic, because you could just tell if it was, killing more cells, essentially. It'd be hard to determine if it was more transmissible, just based on this current study. So it's one of those gray areas about whether you could classify this study in question as being a quote-unquote gain of function study. I myself, I guess, am probably taking the unpopular opinion that I'm going to withhold judgment. So I'm not an expert on gain of function studies, or sort of the I guess the ethics and the politics behind it. So my background is in biochemistry, so I understand the techniques that they're using, but I guess from the sort of more political or even philosophical side of things, I'm not sure if I could be the one to determine whether this was sort of solidly in the realm of gain of function, with all the sort of negative connotations that surround that, or if this was a just sort of standard, recombinant DNA technique that was used. I mean, it is a recombinant DNA technique. I mean, in my own research, I've done things like add the beta lactamase enzyme, which confers antibacterial resistance into E. coli in order to isolate the protein. Of course, the problem with the thing with that is that it would confer, I guess, antibiotic resistance to the E. coli that I'm putting it into, but the E. coli is sort of a laboratory attenuated version of E. coli. It's not pathogenic. It's not pathogenic. It's not dangerous, and it is not transmissible by itself. And so, giving it that antibacterial resistance does not make it any more dangerous, really. But that's what I'm talking about as far as the gray area is. Did doing this experiment, putting this other spike protein into this WIV1 backbone, did that make it more pathogenic or more transmissible? I'm not sure. I don't think it would make it more pathogenic. So because we are not giving this anything that makes it more dangerous, really, I could see that we could make a case that it would make it more transmissible. So, for instance, if the transmissibility that's already present in the WIV1 and the transmissibility conferred by this other spike protein gave it transmissibility above and beyond what those two things in their natural sort of background would be, then you could say that would increase transmissibility. But anyway, I'm just kind of thinking out loud here. I hope what I've done is sort of give some background on this issue, so that maybe you can understand the debate a little bit better, and maybe even come to your own conclusion insofar as whether what this paper was doing was so-called gain-of-function research in the negative connotation sense, or if it was just a sort of standard sort of recombinant DNA technique experiment. And so, like I said, that kind of goes back to how broadly we want to define sort of a negative sense of gain-of-function research, where if we define it too broadly, then we might include sort of standard recombinant DNA techniques that are necessary for pretty much all of the sort of biological sciences and biomedical sciences and things that are necessary for all of the types of things that we know and love about medicine. Or if we define it too narrowly, we might miss some potentially dangerous sorts of experiments. And so that's why I thought that this criteria of novelty, pathogenicity, and transmissibility is a good way to sort of assess whether something is the type of research that we would want to continue funding and to have to continue having being performed in laboratories in the United States and around the world. And so anyway, I hope you found this video useful, and I will see you in another video.